How many people in this room know the game Cluedo? A reasonable number of you. So it's a game that we play a bit in England where the idea is someone's been murdered and you have to work out who's killed them, with which weapon and in which room. So I'm going to talk about Genomic Cluedo today, which is the idea that we have from genome-wide association studies a large number of pieces of evidence that suggest a bit of the genome is influential in disease risk. But what our job is to try and work out which gene that's acting through, in which cell type, under which environment. So that's the link. So, and I'm going to talk about this in the context of multiple sclerosis, which I'm sure you all know is a pretty horrible chronic neurodegenerative disease. All you really need to know about it for the purposes of this talk is that in terms of understanding why people get multiple sclerosis, actually an awful lot is known. We know that it's essentially the result of a dysregulation of the immune system whereby the body starts recognizing bits of the protein surrounding myelin in the brain as foreign and starts mounting a response against it. So we actually know quite a lot about it, and we even know, for example, here there's sort of molecular evidence on how there's an unfortunate mimicry between bits of proteins that a virus that we're pretty much all exposed to called EBV mimics something that's found in the myelin proteins. And so this is, it's not the only, but it's one of the sort of the key, probably one of the key elements driving that autoimmune response. So we know quite a lot about why people get MS. But the question then is, why, if we understand that, can we not treat it very effectively? Now, actually, that's not quite the right thing to say, because multiple sclerosis is a disease that has a reasonable number of quite successful drugs for its treatment. Here's a list of the primary approved drugs for the treatment of multiple sclerosis. They kind of fall into 2 or 3 categories. Some of them are essentially immunosuppressant. They sort of activate bits of your immune system that try and dampen responses. Those tend to not work particularly well. They have a sort of a low response rate. Down at the bottom, we've got the sort of the nuclear options, which work moderately well but have unfortunate side effects. So, for example, that you can stop T cells getting into the brain. That seems to work rather well in terms of stopping getting the disease, but the problem is that about 1% of people get reactivation of another virus that we're all carrying called the JC virus, and that leads to a fatal leukemia. That's not good. And then there's another nuclear option, which is basically to try and reset the immune system by wiping out mature lymphocytes. So those are licensed, or at least one of those is licensed, but they're not massively widely used in the UK. These 2 treatments are probably the most widely used. They're remarkably effective in terms of reducing the symptoms of MS. The problem is they don't actually slow the rate of the underlying disease, so that over a period of time, the disease is getting worse and worse. You don't recognize that, but at some point, the treatment no longer works, and essentially the person has the full disease as if they had been on no treatment anyway. So it's essentially affecting the symptoms, not the underlying cause. So there are lots of treatments. They don't work particularly well. And why don't they work very well? Why don't we have an effective drug? Well, reprogramming the immune system, which is kind of what you'd have to do to completely cure you, isn't really feasible. So what we're looking for are ways of nudging the immune system around such that it has much less of this autoimmune response. And the reason that's hard is the immune system is incredibly complex, incredibly interconnected system, so that we kind of don't know where to start. Now, this is, of course, where genetics comes in, because genetics gives us a really good handle on where in the genome there are these genetic nudges that are pushing our systems towards or away from a disease state. And in MS, like in many autoimmune diseases, we now have well over 100, I think at the latest count is something like 250 bits of the genome, where there is a variant that influences your risk. The vast majority of those we know nothing about the mechanism for, of course. But what we do know is that there's a very substantial shared genetic etiology with many other autoimmune diseases. This is just a PCA of the coefficients, the betas, for various autoimmune diseases. And MS sort of sitting somewhere in the middle of that. So there's a shared etiology which suggests that some bits of the immune system sort of generic up or down, some bits are probably more MS specific, some bits are IBD specific and so on. And so that again gives us another handle into identifying those nudges in the immune system which are specific to MS. What can we do with these data? Well, one way of looking at the GWAS results is to try and do not the fine mapping, the sort of the detailed mechanistic understanding, but what you might call gross mapping. That's trying to understand tissues or conditions in which are particularly enriched in the genetic influence on multiple sclerosis risk. So this is a chart taken from the EBI, which shows a whole bunch of cell types and essentially the enrichment of MS loci in these around enhancers active in these different cell types. And you can see that, not surprisingly, there are a whole bunch of cell types that are sort of enriched for MS hit, and guess what? They're all in the immune system. So this kind of work tells us cell types or bits of our tissue architecture which are relevant for the disease, but they're not actually really going to give us much in the way of mechanism. You know, if I told you that MS is an autoimmune disease, is a disease of the immune system, you're not going to give me a Nobel Prize for that. So what else can we do? So I think that the whole point, essentially, of doing genome-wide association studies and trying to follow it up is that it should give us new targets for treatment. We should be looking for drug targets, which we think, and here I think is the critical issue. I feel I should, as a statistical geneticist, put my money where my mouth is and say, here is a target I think you should go after as a drug company. I think that's what we should be trying to do. But of course, that's not a trivial thing to do, and we need to be very certain, before we make those statements, about where we think the mode of action is. So it's not just good enough to say, here's a SNP that influences risk. We need to understand which gene it's affecting, how it's affecting that gene, which cell type, which kind of environment that's working in. So we can actually make a pretty good start about understanding how you should focus drug treatment to that particular, or drug development in that particular case. So I'm going to give you 3 brief stories about the search for mechanism that I've been involved in. These are all MS related, and they kind of give you a sense of the spectrum of types of things that one can try to do. The first of these is a specific example around a gene called TNF-RSF1A, which is kind of a beautiful case about how you could use genetics to identify drug targets, but kind of completely goes the wrong direction. I'll then talk about the hunt for mechanism by searching for epistasis between genetic factors influencing risk. And then finally, I'll finish up with, I think, where we should all be going, which is the value of data integration, trying to pull together lots and lots of different types of omic information to really pinpoint what's going on. So first of all, this TNF-RSF1A, which it sort of looks like a dream. So GWAS gives us typically a region of association. Often we can't pinpoint that to a very specific SNP. We know there's a region, but we can't say which SNP it is. A few years ago, we developed this machinery that tries to say, of the variants in this region, which one do we think is the most likely to be causal, and how much evidence should we associate with that? If you apply that to the hits that you get in multiple sclerosis, if you just look at the central chart in here, you're kind of interested in the one on the far left, which is how many hit regions do you have a single target in? And the answer is very few, maybe 5 to 10 or something like that across all the known MS loci. So it's not many, it's a very small fraction, but nonetheless, they're low-hanging fruit, they're good to go after. And one of the most striking examples of one of these sort of one-hit wonder regions is this a region in a gene called TNFRSF1A. I'm not very good with gene names, where we could very convincingly pinpoint the risk to a single SNP, which has a substantial effect on increasing your risk for multiple sclerosis. That in itself is quite interesting, but through working with Lars Fugger in Oxford, we were able to work through the mechanism by which that influences risk, and it turns out to have a really interesting structure. So this variant sits in an intron. It affects splicing of the protein such that it turns a full-length protein into a truncated protein. That's interesting because it results in, rather than having a receptor that's anchored in a membrane, the receptor is free-floating. So here's just a cartoon of what's happening. Here's the standard full-length protein. It's a receptor for TNF, a strong pro-inflammatory drug. Sorry, not drug, cytokine. So it normally sits in the membrane. It's a receptor for TNF. When TNF comes along, it trimerizes and activates the downstream signal. This truncated version is no longer anchored in the membrane. What happens is it ends up floating around, free-floating. It still binds TNF, but what it's doing is essentially sequestering TNF. It cannot activate downstream. And it's this sequestering TNF that increases your risk of MS. Now that's kind of surprising because actually TNF antagonists are a very well-known treatment for many autoimmune diseases such as RA. But what is unusual is that if you give people with MS TNF antagonists, which you'd think was dampening the immune system, therefore would be a good thing, actually the disease gets worse. Indeed, if you give people TNF antagonists who don't have MS, they sometimes start to develop symptoms that look like MS. So it's a case where there's a drug which is mimicking what's going on in the body, or vice versa, a natural variant that's mimicking the drug. And in this case, they're kind of doing the same thing. The TNF antagonist increases the risk of MS, both in the drug setting and also in the setting of the natural cells. Now, of course, drug mimicking nature or nature mimicking drug is just what we're really trying to look for, but in this case, it's going completely the wrong way. So in terms of trying to understand places for drug development, this is a non-starter. So that's a sort of a nice example, but as I told you, there are very few places in the genome where you can zoom in on a single variant as simply as that. So how else can we try and identify mechanism? Well, one way we can try and find mechanism is by looking for interactions between different bits of the immune system or different bits of the genetic risk. Here, there's a sort of a very nice precedent for this that has been found in AS and psoriasis, where there's an interaction between a classical HLA allele, this is the psoriasis case, so there's a classical HLA allele CO602, and then there's a risk variant RS30187 that sits in a gene called IRAP1. And the point is this IRAP1 variant only affects risk in the presence of this classical HLA allele. That's a statistical interaction, or epistasis if you like, between these variants. And what's particularly nice about this is that mimics or reflects an underlying biological interaction between these elements of the system, so that IRAP1 is actually part of the scissors that are cutting up proteins in our cells before they're loaded onto the MHC and presented to the outside world. So there's this biological interaction between IRAP1 and the classical HLA loci is reflected in that statistical interaction. So it's a really nice example of how you might use genetic interactions to identify biological interactions. So the question is this a general thing? Can we find lots of examples of this and use that to try and pull apart what's going on? So we've been trying to do that in multiple sclerosis. We developed ways of imputing classical HLA alleles so that we can work in the cohorts of many hundreds of thousands or many tens of thousands of individuals that we need to get signal. The imputation works pretty well. I won't go through that in any detail. But essentially what we can do is build up a model or an architecture of how genetic variation within the classical HLA region affects the risk for multiple sclerosis. So we've got a fairly rich description of many things affecting risk, and we understand many of the classical loci and many of the other things going on there as well. So we have this sort of pretty detailed model for MS risk arising from the HLA, in which you get a series of HLA class 2 risk alleles and a series of HLA class 1 protective alleles. So it's a very nice, simple asymmetry. We don't quite understand the cause of that. And we see various departures from additivity. Importantly, though, are we finding interactions? So what we can therefore do is to take these HLA regions and ask, for all these 100 or so other bits of the genome that we know light up as influencing risk for MS, do any of them show interaction with these classical HLA loci? And the short answer is no. Absolutely none. That's a QQ plot in the middle. This shows sort of the evidence for interactions, and if there were, you'd see these points going up off the top, and we don't. And we don't for any of the classical HLA allele. We've looked at it in lots of different ways, and we don't find interactions. The only interaction we do find, which is actually quite an interesting one and quite strong, is that there is actually an interaction between two sets of HLA class 2 alleles, which we think probably reflects some kind of cross-reactivity with epitopes. So there is some very little interaction, but basically there's a very sparse landscape of interaction. So in terms of helping us define mechanism, that's probably a no. It's unfortunate, but that seems to be the case. One final thing we did was to ask, well, maybe we're not looking at it in quite the right way. Maybe we could find ways of trying to look for evidence for what you might call polygenic epistasis, whereby the broader genetic background affects the extent of risk of a particular strong classical HLA allele. And so we developed a method for doing that, which I won't go into in any detail. Basically, the sort of the observation is that epistasis induces LD within cases that you don't see in controls, and so we can test for that. And again, guess what we find? Basically no evidence for this kind of polygenic epistasis either. So there's been a lot of discussion about epistasis and whether that might be an important contribution to the missing heritability. Our experience, at least in multiple sclerosis, is that there's absolutely no evidence for it whatsoever. So end of the line for that idea. That's a shame. So where can we go? So the final thing that we might try and do, and it's the last thing I'm going to talk about, therefore it's probably the most likely to succeed, I wouldn't have put it that way, is this thing about integrating the multiple data sources, and in particular trying to bring together the very rich data sets on gene expression, chromatin state, and so on, that are being collected not by many different groups all over the place, and not just in one tissue, in lots of different tissues, and not just in lots of different tissues, but in lots of different tissues under lots of different environmental challenges. And I think it's that last thing about the sort of heterogeneity of environmental challenges which is going to be really key here. So I'm going to talk about a specific region. It's another region I've worked on with Lars Fugger in Oxford. It's around this gene called TYK2, tyrosine kinase 2. Kinase is sort of the key thing there. So it's involved in a sort of a signaling response. But it's a region, it's one of these regions that you get a few of in the human genome where there are an awful lot of variants in this region that have been described as associated with genetic risk for lots of diseases. So not just MS, but lots of other autoimmune diseases. And so there's this sort of rich description of association. And what we wanted to do was to try and work out what the actual underlying architecture was and how that was translating into, or how that genetic architecture translated into molecular or mechanistic architecture. So what we did was sort of a Herculean effort by people in my group was to actually get together at the individual level, large numbers of, well, here 5 different case control cohorts. Actually, we got hold of the genotype level data through begging, borrowing, and stealing. And the short version is, within this region, we think there are 3 genetic variants that affect risk for multiple sclerosis and other autoimmune diseases. The top hit of these is actually within the TYK2 gene. This variant is in the kinase domain. It's a non-synonymous change. It abolishes, or it's a pretty substantial change to the kinase domain. You'd think that that might be a pretty good candidate. So this is one of these sort of one-hit wonder regions where indeed there's only a single variant that it maps to. This has an extremely strong protective effect across a whole slew of autoimmune diseases, less of a protective effect in a heterozygous state. This is fantastic. This is, from a sort of trying to identify drug target point of view, this is just what you want to see, a very strong protective effect that doesn't have any substantial pleiotropy, i.e. it's protective in all of these cases. There are, however, other signals. There's a second signal over here. This is around a gene SLC44A2. This is only found in multiple sclerosis. There's some sort of weak evidence for it being involved in the other ones. But you can see it really sticks out in MS and nothing else. So that's the second signal. And then the third signal is, again, nearby this TYK2 gene. For a long time, people thought that there's another non-synonymous mutation in TYK2, which is in something called a pseudokinase domain. And they thought that that was probably the lead SNP. Actually, we're now convinced that that's not the one. It's actually, it's a mutation that's a bit downstream of that, and it affects ICAM1, which is a cell adhesion molecule. And this shows a different pattern where it's risk in some autoimmune diseases and protective in others. So just within this short region, you've got the sort of whole microcosm of different types of genetic architecture between these variants and risk for autoimmune diseases. So that's nice, but we obviously then want to go on and look at the functional, the mechanistic side of that. So where do we get to? So if we just look at this TYK2 variant, we've looked at the kinase activity in vivo settings and ex vivo settings, and basically across a whole load of immune cell types, we see strong reduction in that kinase activity of this variant, of the protective variant, which is just kind of what you might think. But again, it's sort of really good confirmatory information that this really is the mechanism by which that is acting, and therefore is a good candidate for pursuing as a therapeutic target. What about the other regions? Well, nothing else is quite so barn door, but what we can do is to bring together, as I said, all these EQTL data. This is a map of the EQTLs in this region for all the genes. So here are the genes. A blue line indicates an EQTL, and there are 3 different cell types that we're looking at with 3 different stimuli. Okay? And what you can see basically is that every gene in this region has an EQTL, which is, I think we heard yesterday how as you start compiling these big data sets, basically every gene that you look at now has an EQTL somewhere. So having an EQTL isn't special. What is turning out to be special is when that EQTL is active. So we can try and integrate the genetic and the functional genomic data here and ask, for example, this second SNP, which was the MS-specific SNP, that is in the region of this gene. This gene has the SLC gene. The SLC gene has a load of EQTLs. Is this genetic risk factor the same thing as the EQTL? And the answer is no. They are distinct signals. So that's nice. We've been able to show that actually that EQTL isn't the mechanism by which this risk is being delivered, if you like. And in fact, we think it's a gene somewhere over here that's being affected. So you can actually use the eQTL data to really refine your hypothesis about what's going on. What about that third signal? Well, the third signal turns out to be very interesting. It's over here, so this is, as I said, around this pseudokinase mutation, but there are other things nearby. So it's not an eQTL for TIC2. TIC2 only has an eQTL in another part of this region. Interestingly, actually, the kinase domain mutation, which we know is causal, isn't an eQTL for anything, as far as we can tell. What about this? Is this mutation associated with any of the eQTLs at all? Well, the answer is yes. And it's rather nice in that this causal mutation over here is associated with an eQTL for ICAM1 that is only manifest when you stimulate monocytes with LPS. So it's an environment-specific eQTL that only appears once you sort of kick the immune system into activity. And that's obviously very nice because it's helping us identify the cell type and the condition under which this variant is being affected. And so now, of course, we want to then understand how changing ICAM1 levels are actually affecting the downstream process. So that's basically all I want to say. There are a few sort of take-homes from this. One is that there's a whole bunch of statistical tools coming out for dealing with these larger data sets, which are useful. This fine mapping and the co-localization work, the methods are pretty sensible, pretty simple. Most of the work is actually to do with accessing the data and making sure it's all normalized and harmonized well. I told you a bit about this search for polygenic epistasis. I thought it was a really nice idea, but it turns out to be pretty useless in the case of MS. The one thing I would say is that we do find that having access to individual level data is hugely valuable in doing this work, and that goes back to the discussions yesterday about data sharing and data access. From the biological perspective, this sort of finding of a very sparse landscape of interactions influencing MS, I mean, it's only MS I've told you about, but my guess is it will pan out in other diseases as well. We find loads and loads of cis eQTLs, or rather, other people have found loads and loads of cis eQTLs, but actually very few of them seem to be linked to disease risk. So really, what's important seems to be the cell type by environmental exposure that is completely key, and therefore the real value in doing that work on a sort of broader scale. And then from the context of MS, do we have new targets? Well, I think that TIC2 is a really good drug target. I know Merck is very interested in that, and there's another company also very interested in that as a way of building a therapeutic intervention. This result about the interaction between the class 2 alleles does suggest some degree of cross-reactivity, and that might be worth pursuing further in an animal model. So that's where I want to stop. I just want to thank all these people, in particular Lars and his group, and Luke and Adrian, who did most of the work on the TIC2. Thanks.
Thanks very much, Gil. That was brilliant. Are there questions for Gil? And if you could come to the microphone, that'd be helpful.
Thank you. That was a great presentation. I have a question regarding your last slide on this lack of correlation or lots of eQTLs, cis eQTLs, but few linked to disease. Is that maybe because we haven't looked at the protein downstream? So, I mean, how well does cis eQTL correlate with protein expression? Do we know anything about that? And if you integrate that kind of data back into your model, will it reveal something?
So we don't have data on protein levels, and so I can't answer that question. If you don't affect the, so these clearly are going to affect the level of expression. Will they affect protein levels? Who knows? It's unlikely to have something that affects protein level, less likely to have something that affects protein level that doesn't affect transcription level, but you know, of course there are ways around that. It would be really good to get those data. I think the sort of the kinase domain SNP is actually a really interesting case where that must be affecting the expression or the activity of a whole slew of proteins. And we can't see it in the data that we've collected, or what other people have collected, presumably because it's just quite subtle and you have to look across lots of genes simultaneously. It's a sort of a pathway response rather than any single gene being having a huge effect. I think trying to use that as a positive control and understanding how you can find that kind of cellular pathway response from high throughput omic data sets, whether it's expression or proteomics or even sort of imaging data or something like that, I think is a really interesting question.
Thank you.
If I may. I have a question. I thought it might be fun if you might comment on epistasis and well, at least in this case, you weren't able to capture a signature for it in the context of MS. There have been some recent publications which have claimed that there's been a signature genome-wide in general. Care to comment?
So there's epistasis that you need to measure an endpoint. And here, obviously, we're measuring a disease endpoint, and we're not finding huge evidence for epistasis. Most of the work I think you're referring to was more about gene expression and sort of interactions there. Some of that work has been criticized already and pretty compellingly shown that it's not epistasis, it's just a lack of information. One of the things that we really found in doing the epistasis work was you have to be, I mean, it's a trivial point, but you kind of have to be looking at the right variants to not see epistasis. And if you look at the wrong variant, you can apparently see epistasis because of the fact that, the haplotypes are capturing the true causal variant that you haven't measured. So I think that the conclusion of epistasis is one you have to be very cautious about because there are lots of ways, and we fell over a couple of times before we got to this point, there are various ways in which a simpler architecture can look like epistasis just because you're not measuring everything that you should be measuring.
So in relation to that, and the fact you did capture something of a gene-gene interaction with the HLA classes, which wouldn't necessarily classify that epistasis, but it made me think about whether this was, with HLA, of course, you have allele frequencies that are more intermediate, and you have potentially more power. So is there a question of power as well? You mentioned endpoints as being critical, of course, but...
There's clearly a question of power. We've done quite a lot of power simulations to look at the polygenic epistasis, and we are totally confident that were polygenic epistasis explaining as much variation as the main effects, we would be very well powered to see that. So I'm not going to completely rule it out, but we can put an upper bound on it, and it's clearly much less important than the main effects themselves. So we're never going to be able to say it doesn't exist, and it's clear from a lot of model organism work that it can be, there is epistasis, it can be expressed. What I think we're saying is that there's not facultative epistasis in terms of the endpoint we're measuring in the natural human population. And for me, that's what matters, I think.
One last question?
Yeah, I have one more question. In your opinion, do you think it is possible, given the temporal, the importance of the temporal expression for EQTLs, in the context of population, how do you capture that? When you're doing baseline, people are at all kinds of different time points in the development of disease. Can you capture that? And how do you translate that from the cells to the
that's
population?
A really thorny question, I think. You want to measure everything on everyone, and you can't. I think so. There are two approaches to that. One is you kind of assume by sampling lots of people in lots of different time points, you're kind of capturing that heterogeneity. So as long as you can capture the information that points you to the state at which those individuals are in and use that, you should be able to reverse engineer, essentially. The alternative point is to say, well, I'm going to do a much more controlled thing. I'm going to take individuals who are much more homogeneous, and then I'm going to take cells from them and do things to them. Clearly, the latter approach is what people have done so far largely and is more likely. There's a clear path to success there. The former is very exciting and has a lot of potential, but we need some good positive controls, I think, from that. You need to take cases where you know what you're looking for, see if you can do it in that basically random sampling approach, and work out the sample sizes you might need and the degree to which you've got to capture the ancillary information. I think it's a hard call, personally.
Any more questions? No? Well, thank you very much.